So the topic for today is on mutations. There's going to be two different ways, well, several different ways of describing mutations. But anyway, okay, so we started the chapter by talking about cystic fibrosis. Cystic fibrosis is a result of a mutation. A mutation is a change in the nucleotide base sequence of a gene or DNA molecule. Now, one way to classify mutations is what kind of cells are affected by mutations, and that has an effect on either the organism or future generations. A germ cell mutation happens in gametes. So another word for gamete is germ cell. This is sperm and egg. If a mutation happens in the germ cell, it's not going to affect the organism itself, but it can be passed to offspring. The other kind of cell is the other kind of mutation is somatic cell mutations. Somatic means body. Actually, soma, the prefix soma means body. So if you have a mutation in your body cells, it will affect, or I should say it can affect the organism, but it cannot be inherited because it's occurring in just a regular body cell and not in a reproductive cell. So an example of a somatic cell mutation is types of skin cancer and leukemia. A lethal mutation causes death, and this often happens before birth. So if there is a lethal mutation in an embryo, then often it will result in what's called a miscarriage, which is when the fetus is spontaneously aborted, meaning because there was a lethal mutation, the fetus cannot develop properly, and so the body gets rid of it naturally. Now, sometimes there can be beneficial mutations. Some mutations result in phenotypes that benefit the individual. Benefiting meaning that they have a better chance of surviving and reproducing. So this is going to have an evolutionary advantage. So if you look at mutations from an evolutionary point of view, like in the context of will this help the species survive better in the future, that is what natural selection is all about. That's something we're going to discuss probably grade 12 is where I've slotted that topic for, but it comes up a lot in other kinds of chapters, because when you talk about mutations, when you talk about adaptations like sexual reproduction versus asexual reproduction, it's all a matter of natural selection. So natural selection is a process where individuals are either well-suited or ill-suited to their environment. That ends up resulting in either they die, which means their genetic makeup, which is not suitable for the environment, is taken out of the gene pool. The gene pool is basically all of the genetic information that's available for future generations. If you have a mutation that is beneficial, it makes an individual stronger or faster or whatever the adaptation or the mutation happens to result in. If it makes that individual better suited to survive the environment, it's going to survive and reproduce and pass on that mutation. If you have a mutation which is maybe not lethal, but it puts you at a disadvantage, then you probably won't succeed in reproducing, which means that bad mutation does not get passed on to the next generation. So this process is occurring all the time. And as it occurs over generations, you can see the traits of populations actually changing. For example, the human appendix. The appendix is basically, you've got your large intestine, which basically goes like that. And your appendix is pretty much just kind of like a little organ that shoots off of your large intestine. It has no purpose, no function whatsoever. What it is the leftovers of an organ that used to be useful in our ancestors. So it does give us insight as to like where we come from as a species, but we are in the process of evolving the appendix out. The only time that it affects us is when it gets infected and inflamed, and that is appendicitis. If you're planning on becoming a doctor, itis means inflammation. So anytime you see itis at the end of a word, it's the inflammation of whatever the first part of it is, so appendix. And it can result in the appendix bursting, which results in you dying. But normally the pain is very, very excruciating, and so you end up in the hospital before that happens. Okay, so that's basically how it works. All of the genes, whether they are a result of crossing over or mutations, the genes that are favorable, that allow the species to survive better, those genes will be passed on to new generations because they allow the species to survive more successfully. All right, so we have germ cell mutations and somatic cell mutations. Now we're going to divide mutations into where they occur. So we have chromosome mutations. Obviously, these are going to occur on chromosomes. They're going to involve changes in the structure of the chromosome. So we're not talking about nucleotide bases yet. So you can either, depending on the kind of chromosome mutation, oh, okay, Bluetooth. Depending on the kind of chromosome mutation, it can either result in exchanging information between two non-homologous chromosomes or a loss or gain of pieces of chromosome. So the first chromosome mutation is called deletion. Obviously, this is a loss of a piece of chromosome due to breakage. So in this example, we have completely lost that entire section of the gene of the chromosome. So none of those genes will be passed on. Again, it depends on what chromosome this occurs on, how big the piece is, and what information was contained in those genes. The second kind of chromosomal mutation is called inversion. Basically, you have a segment of a chromosome that breaks off, flips around, and reattaches. So how that affects inheritance is that the gene order you can see has been completely flipped around like that. And so if crossing over occurs, this could result in a greater genetic variety because you're changing the order of the genes. So different genes can be exchanged between homologous chromosomes. Translocation is where a piece breaks off and reattaches to a non-homologous chromosome. So normally during crossing over, so like inversion, you would have like chromosome number 4 and it's still chromosome number 4, but let's say when it goes to crossing over, you've got the homolog. So I'll just call it 4'. So during crossing over, homologous chromosomes do a synapse, right? And they can exchange information, but they carry the same genetic information, meaning like whether it's eye color or whether you're right-handed or left-handed, whatever the genes are, they contain the same genes, but not necessarily in the same form, if that makes sense. So you can call this like mom and dad. But the point is crossing over occurs between homologous chromosomes. In translocation, you can see here, we've got chromosome 4, chromosome 20. They don't have the same genes at all. They contain completely different genetic information. So let's say, normally this is like, let's say this is the gene for eye color on chromosome 4. Oh wait, that's not the one that got exchanged. What got exchanged? Here, this one. Oh yeah, and the green one, these two got swapped. So if this is the gene for eye color, it went from chromosome 4 to chromosome, oh wait, what is it? No, I'm wrong again. Oh my God, these colors, I hate these colors. All right, so chromosome 20 does not have red at all, chromosome 4 does. So this piece of chromosome 4 broke off and got attached to the bottom of chromosome 20 right here. There it is. So this piece got inserted right there. Yeah, because then you're left with purple, green, orange, blue, purple, green, orange, blue. Yeah. Wow, they could have really used better colors. Anyway, so now whatever this genetic information is here is no longer located on chromosome 4, it's located on chromosome 20. That might be a problem because now you don't have an alternative form on the homologue for chromosome 4 and chromosome 20. So if there's any kind of recessive genes there, then they might end up coming through because there's no allele on the other homologue. And I think the last chromosomal mutation is called non-disjunction. This is where a chromosome fails to separate from the homologue during meiosis. So what that means is basically when the chromosomes are supposed to split, one of them doesn't split. So you can see during meiosis 2, so during meiosis 1, you've got the chromatids split apart, right? So you've got one half of this chromosome and one half of that chromosome, and same with the red ones. And here you got the same here. So that split evenly, right? But during meiosis 2, you can see you're supposed to end up with each cell having one red, one blue. So red, blue, red, blue. This one had only red. So these two blue chromatids failed to separate. So you've got one cell that has n-1, so it's missing a chromosome, and this one has an extra chromosome. We see this in a condition called Down syndrome. That's when you have, if it's a female, they have XXX. If it's a male, XXY. So the X chromosomes failed to separate during meiosis. Now we get to what are called gene mutations, okay? So we're inside the actual nucleotide sequence in DNA, and these are going to be called point mutations because they are a single nucleotide that's being changed inside of a gene. So we're no longer talking about chunks of chromosome, we're talking about a specific nucleotide in the nucleotide sequence of DNA. This goes back to chapter 10, talking about protein synthesis and codons and how the sequence of amino acid translates to a sequence of amino acids, which translates to the protein. So one example of a gene mutation is called substitution. This is where you've got one nucleotide that replaces the other. If this occurs in a codon, then the amino acid can completely change. So you can see the original sequence, You can see the original sequence is supposed to be CTT on DNA, which means the mRNA transcribes to GAA because G pairs with C, A pairs with T. GAA is the codon for glutamic acid. And a substitution is going to be this thymine being replaced with adenine. So now you go CAT, the mRNA becomes GUA, and this codes for valine. So now you have a completely different amino acid, which means either you're going to form a different protein or you're going to form something that just can't form into a protein. It doesn't work. So what the protein you're trying to make, it's abnormal, it doesn't work. Then you have a deletion. A deletion is where one or more nucleotides is lost. This is going to end up in something called a frameshift mutation, because if I've got, let's say, GACCATT, okay? So that would transcribe to CUGGUAA. So my codons would be, I might have to add some in here. I'd have CUG, GUA, let me add a CC, that's fine. So I get 3 codons, GG, okay? So what happens if this C is eliminated? So now I've got CUG, oops, yeah, UAAGG. So now I've completely changed, I've got CUG, that still stays the same, but next is UAA. So everything that comes after that has been shifted. So all of the amino acids after the deletion are going to change. The same thing happens during insertion. Insertion is where you have a nucleotide that's added to a gene. So TTAC. So that is going to transcribe to CUAAUG. So you've got CUAAUG. If I add a C in here, then I've got CUAGAUG. So I've got CUAGAU and then whatever comes afterwards. So you can see how that completely changes. The first one's still okay, but after that we go from AUG to GAU and everything else changes. So here's what the normal gene sequence or the normal base sequence looks like. During a frameshift mutation, you can see that we've eliminated or we've deleted the G. So I like this because I want you to see how what the frameshift mutation looks like. So if we've got ACG, CTC, GTC, and then the mRNA, UGC, which is cysteine, GAG, which is glutamic acid, CAG, which is glutamine. If we remove that G, we start with ACC, which transcribes to UGG, which is tryptophan, completely different. You see that the entire amino acid sequence is different now because of the frameshift mutation. For a substitution, you're just substituting for that one particular codon. My Bluetooth keeps disconnecting. Strange. Okay.
What do you think of when you think of the word mutation? Do you think about X-Men and some really awesome ability? Or the Ninja Turtles? We loved the Ninja Turtles when we were little. We might be showing our age a little bit. But mutations really are not that glamorous. Most people understand that a mutation is some kind of change. It is a change of genetic material, and more specifically, a nucleic acid. RNA and DNA, they're both types of nucleic acids. So how does a change happen? Remember that in DNA, the base adenine A goes with the base thymine T, and the base cytosine C goes with the base guanine G. And that's all good, but what happens if the wrong base matches up? Many things can cause an error like that. There can be external factors, like chemicals and radiation, internal things too, like something goes wrong during DNA replication, during interphase. Interphase is a stage that prepares cells to divide during mitosis. And now while these things can increase the chance that a mutation is going to happen, it's important to understand that mutations, they're random. If a mutation is going to be a helpful thing for an organism, which is rare, very few mutations are actually helpful, it can't just will itself to get that mutation. An organism can't just will itself to get this. It's definitely not like X-Men either. And more about that when we get to natural selection. Many mutations are actually neutral in their effect, meaning they neither help nor harm an organism.
This would hold especially true if the mutations occurred in the introns. If you remember introns and exons, exons are the ones that have the codes for the amino acids, the introns don't. So any mutation that occurs in an intron won't have an overall effect.
And some mutations are harmful. So we're going to talk about the different kinds of mutations. First, we're going to talk about gene mutations. So DNA makes up genes, and genes code for proteins that influence or make up different traits. So when DNA has changes, otherwise known as a mutation, then different proteins can be produced. and this can affect an organism's traits. So let's look at the gene mutations. So first, substitution. That means you have the wrong base matched. So instead of A with T, you could put A with G. Scandalous. You can have insertion. That means an extra base is added in. You can have deletion. That means a base is removed. Insertions and deletions have the potential to be especially dangerous because remember, in protein synthesis, we talked about how bases are read in threes. Well, if you add a base or remove a base, suddenly the number of bases total has changed, right? So if you read in threes, depending on where it happened, everything that's read afterwards has the potential to get really messed up. We call this a frameshift. Now, these were all types of gene mutations, but we also have something called chromosome mutations. Remember that chromosomes are made up of DNA and protein. They're highly organized, and they have a lot of genes on them. So all of the body cells in your body have 46 chromosomes. Human sperm and egg cells have 23 chromosomes. Well, changes can occur at that large chromosome scale too. So let's talk about these chromosome mutations. Just like insertion in a gene mutation where bases got added, you can also have something called duplication in chromosomes. These are mutations where extra copies of genes are generated, so extra copies of that chromosome are generated. There's deletion. That's where some of the genetic material from the chromosome breaks off. Inversion. That's when a broken chromosome segment gets inversed. That means it gets reversed and put back on the chromosome. Or translocation. We were not kidding back when we said there's a lot of trans words in biology. That's when a fragment from one chromosome breaks off and attaches to another chromosome. There's more mutations than what we just covered, but the idea here is there are many kinds of different changes that can happen. If a mutation is going to happen, there's also some vulnerable times, like when DNA replication happens during interphase, and also other times too, like meiosis. Remember, in humans, meiosis produces sperm and egg cells that have 23 chromosomes. But sometimes those chromosomes, when they're separating, they don't separate completely. We call this non-disjunction. This results in an egg or sperm that has too many or too few chromosomes, and that can cause a genetic disorder depending on which chromosome we're talking about. Different chromosomes contain different genes. So the specific chromosome that's affected, that really does make a difference in the result. And let's talk about some real-life examples now of mutations. Remember, we said that a lot of times mutations can have a neutral effect. Not all of your DNA codes for a direct protein. And other genes in your body can even influence whether those genes are going to be turned on or turned off. Let's talk about sickle cell anemia. First, a little background. Hemoglobin is a protein in your red blood cells that helps you carry oxygen. Well, in the disorder sickle cell anemia, the gene that codes for hemoglobin is mutated. And if you inherit 2 copies of this mutated gene, one from each parent, you can have this disorder. And the disorder makes it very difficult for your red blood cells to carry oxygen because the shape of the red blood cell, it's affected from this mutated hemoglobin protein. And this can lead to anemia and other problems. But get this, let's say you just inherit 1 copy of the mutated gene from one parent. You're going to be considered a carrier, but you don't really officially have the disease, and usually you don't even have symptoms. But what's interesting is carriers appear to have a protective factor against malaria. Malaria is a disease caused by a protist that can be transmitted by mosquitoes. These individuals, they can still get malaria, but usually their symptoms are less severe. So in a way, this 1 copy of a mutation can be an advantage if you happen to live in an area where malaria is really present. Studying mutations and genetic disorders is a huge field right now. If this kind of thing interests you, there are a lot of careers to look into. In fact, there's a career known as a genetic counselor. They work to help families that may be at risk or affected by genetic disorders. They do a lot of good in the world. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.